Let us look at the next subtopics here: polymerase chain reactions and gel electrophoresis as tool for amplifying and separating DNA. So we start with the first technique here. There are two techniques: polymerase chain reaction, the first technique; gel electrophoresis is the second technique. So these two techniques are used to amplify. Amplify means to make more DNA. Gel electrophoresis is to separate DNA. Okay, so from here we understand something. The first, polymerase chain reactions is to amplify DNA. So that means is to make more DNA, and gel electrophoresis is to separate DNA. So we will learn each of these one by one. So we start with polymerase chain reactions. We can call it PCR. So PCR is an automated method of DNA replication. So when I say automated, that means we use machine. We use machine to do it. Okay, so it is not happens inside the cell. DNA replication is not happen inside the cell, but DNA replication is happen in the machine. Okay, so DNA amplification, like what I told you just now, make more DNA. The process of producing more DNA with a specific base sequence. So we produce more DNA for certain base sequence. Depends on the reasons that we want to do it. Then we produce certain part of the DNA in a larger quantity. Now the next one, like what I said just now, to do PCR, we use machine, and the name of the machine is thermocycler or thermocycler. So this is the machine, the name of the machine. So the machine looks like this, as you can see in these pictures. We can't see much based on these pictures itself. So let us understand more by looking at the sentence here. This is a PCR machine that follows a cycle of steps repeatedly, doubling the quantity of DNA with each cycle. So that means this is the PCR machine. You put some samples of DNA in the machine, then they will keep making more and more DNA. In general, it works in that way. And after that, thermocycler can amplify multiple samples simultaneously in arrays of wells. So that means you can put a few samples in the machine at the same time, and they can make more DNA for each samples at the same time. Okay, so that is where a machine which is more efficient, as you can see here. Okay, so after that, over 100 in larger machine. So a larger machine can make 100 samples and make more DNA at the same time. Next one, PCR cycle. So of course, you know just now the name of the machine is thermocycle. So it's related to heat and it's related to a cycle. So that's why when we want to understand how they make more DNA, so of course we have to understand the cycle and how the temperature is involved. So we are focused on these two things: how the cycles of PCR works and how the temperature is involved in the PCR cycle. So the steps in the PCR cycle are triggered by changes of temperature. So we look at the cycle, start from number 1. It involves temperature. So let's take a look. Heating to 95 degrees Celsius for 30 to 60 seconds, break the hydrogen bonds that hold the DNA strands together, but does not break the covalent bonds in the two single strands of DNA that are produced when DNA melts at this temperature. Taq DNA polymerase is not denatured by this high temperature. Okay, so we have a few things to understand here based on all the information given. So the first thing is temperature is set at 95 degrees Celsius. At this high temperature, that break the hydrogen bonds. What hydrogen bonds are broken? So we have to remember a DNA strains is made of two strains, and there are base sequence that hold each strains by hydrogen bonds. Okay, so we have to know that. Okay, so they are breaking the hydrogen bonds. They are not breaking the covalent bond. That means we have to know that in each strain there are the phosphate and the ribose sugar that are linked together by using the covalent bond. This bond is not broken. Of course, the temperature is not high enough to break the covalent bond, but it is high enough to break the hydrogen bond. So that's why at the end you can see the pictures. The two strains are separated because the hydrogen bonds in between them are broken, as the base sequence, the complementary base sequence between the two strains have hydrogen bond between them, right? So those are broken. Okay, and then the strains are hence separated like this. Okay, so of course we have to know why they want to separate. We want to make more DNA afterwards. So they also tell us more information like here, Taq DNA polymerase. So the Taq DNA polymerase is an enzyme from a certain organism. The organism is called Taq. So we will learn Taq afterwards. A certain organism supply DNA polymerase to the PCR thermocycle, and these enzymes is a bit special. Normally, what we know is enzyme denature at maybe about 50 degrees Celsius. But at this 95 degrees Celsius, this Taq DNA polymerase is not denatured. So this is the organisms where the DNA polymerase can withstand high temperature, and they can work at high temperature. Okay, so that's the special part. Of course, what is the functions of this DNA polymerase is to make the DNA strains, which is known as the polymer. Okay, now the next one, step 2, annealing, cooling to 54 degrees Celsius for 30 to 60 seconds. Allow primers to bind. Okay, so primer bind to one part of the strain, and then of course primer means if primer go to one strain, and then after that they will make the DNA strains to become longer. Okay, so primer must start first. Primer must go to the DNA strain as the first step before they make the DNA strain longer. So primers. Different primers are needed for the two separated strains. So we use different primers. So the primer will go to certain parts of the DNA strains, and then after that start making the DNA strain. A large excess of primer ensure rapid bindings. So we put more primers in the machine, and then the primer will start the strain. Okay, after that, preventing the single strands of DNA from pairing up again. So they are trying to say that we use more primer so that the primer can stick to the single strand rather than letting the two strands pair up again by using hydrogen bonds. We don't want that to happen. So that's why we use more primer. Okay, now the next steps, elongations. Heating to 72 degrees Celsius provides the optimum working temperature for tag DNA polymerase, which binds to the single-stranded DNA adjacent to the primer. Okay, so basically we have to understand in a DNA replication, first the two strands have to separate by breaking the hydrogen bond. Second, the primer must stick to certain part of the DNA strand, and after that the DNA polymerase have to come to it, go to the primer, and then after that they will start to add more and more nucleotide, and from there the DNA strands is made. Okay, so primer, then DNA polymerase, then the nucleotide come, join up with the primer, and more nucleotide come and join with the primer again. So the DNA strands is getting longer and longer. So that is how it works basically. So let's continue. The tag DNA polymerase, which bind to the single-stranded DNA adjacent to the primer. In 30 to 60 seconds, it assembles new DNA strands. So you can see it takes a very short time to make a strand. DNA polymerase, so works quite fast. Tag DNA polymerase adds about 1,000 nucleotides per minute, a very rapid rate of DNA replication. Okay, so that's the whole process, and this is one cycle. Okay, so from this cycle, we can make a conclusion. First step, break the hydrogen bond. Second step, primer. Third step, DNA polymerase. Go to the primer and add nucleotides to it, and the DNA strand is formed. Just like that. Okay, now PCR components. So in the PCR components, you can see a few things. DNA sample have to put in the machine so that we can do DNA replication. We also need to put primers to the machine so that the primer will stick to one of the DNA strain when the DNA double strands are separated, then primer will stick to it. Then nucleotides, you have to add a lot of nucleotides into the machine also, so that after the primer stick to one of the DNA strain, the polymerase go to the primer, and then the nucleotides will start coming to join one after another, based on the idea of base complementary. Okay, then we have the tag polymerase. So this is the enzymes that make the DNA strain. Then we also have mixed buffer. So when you hear the words buffer, that means they are trying to resist any pH change in the solutions. So they want to maintain the same pH so that the DNA replication can happen in a constant pH environment, as well as the PCR tube. Okay, so the PCR tube, we expect PCR tube is the one that you put everything inside. So put everything into the PCR tube, then they will make the DNA replication inside the PCR tube. Okay, so this machine is expected to have a heating element that heat up the samples into different temperature, and that's why thermal cycle. Okay, so let's see the explanation one by one. Matrix of DNA-based sequences can be loaded into the PCR machines, but only selected sequences are amplified. Okay, so that's the thing. At most of the time we don't make DNA. We don't make the whole strands of the DNA. So maybe we just make certain part of the DNA. Okay, so depends on the purpose. Now, after that, number 2, the optimal length of DNA for amplification is around 100 base pairs. Okay, so that's the optimal length. So that means this machine make a short strands of about 100 base pair. Okay, so primers added at the start of cycling select which sequences are copied. So that means what? That means you can decide what primers to use, and then the primers you go to a particular location of the DNA strain and make a copy of the part of the DNA strains. So of course, try to imagine something like that. If you use primer 1, then the primer 1 will go to one part of the DNA strains, and then they make just a small part of the DNA. Okay, if you are using another primer, so we expect the other primer to go to a different location of the DNA strain, and they will replicate different parts of the DNA strain. So it depends on purpose, like what I said. So that's why we can select what primers to add into the PCR tube so that we can replicate different parts of the DNA strain. So a primer is a short single strands of DNA designed to bind to the DNA at the point. What point we can decide, so you can put different types of DNA. You can put different type of primer, then it makes different replications of the DNA. So where the selected base sequence for replication begins. So it depends on what point the primer is added, and then the replications will be different. The ideal length for primer is 18 to 30 nucleotides. Okay, so from here you can see primer has 18 to 30 nucleotides. Then the PCR make the polymerase. Okay, the DNA polymerase make the DNA strains of about 100 base pair. So altogether we can see how long is that? So it's around 100 to 130, the length of the DNA strands made by the PCR. Okay, will be about that length. So tag DNA polymerase and DNA nucleotides with each of the 4 bases are needed for PCR. So of course we need that. Okay, so I already explained that. Okay, let's keep moving. Tag DNA polymerase evolve. So this is where the tag DNA polymerase come from. Okay, so we have to look at it and understand why it doesn't denature at high temperature. Okay, so reason is here. The tag DNA polymerase evolved in the bacterium Thermus aquaticus. It's a bacteria. These bacteria produce this tag DNA polymerase. So the tag now we understand already. The T, of course, Thermus. The AQ is the aquaticus. So it's the name of the bacteria. And this bacteria has DNA polymerase inside it. And what is so special about this bacteria? So you see here, which live in hot springs. So it can survive in hot temperatures. So that's why its DNA polymerase can be used in the thermal cycle, which is under high temperature. Okay, so including those of Yellowstone National Park, the hot spring over there, Yellowstone National Park, the temperature of these springs can be as high as 80 degrees Celsius. Okay, and these bacteria can stay there. So that means its DNA polymerase can work under high temperatures. So the scientists take the tag DNA polymerase from these bacteria and then put it in the machines for making DNA. So that's how the scientists do it. Okay, so they are not using the DNA polymerase from human, so they are using the DNA polymerase from bacteria, these special types of bacteria. So enzyme in most organisms would rapidly denature. So that is the problem, So we don't use the DNA polymerase from human because at high temperature, the DNA polymerase from human denature. But those of T. aquaticus are adapted to the high temperatures. With the help of tag DNA polymerase, PCR allows the productions of huge number of copies of a selected base sequence in a very short time. Okay, so that's why we use tag DNA polymerase. They can withstand high temperature and they can do the DNA replication very fast. Okay, so I have some question here. Let us skip the question first. And the answers are here. Let's keep moving. Okay, now the next technique, electrophoresis. So electrophoresis compared to just now the first technique is called PCR. PCR is to amplify DNA, to make more DNA. Amplify means make more DNA, but you have to use the words amplify DNA. Okay, so now this is the second technique. The second technique is electrophoresis, which is used to separate the DNA molecules by length. Okay, so let us understand it. Number one, gel electrophoresis is used to separate DNA molecule by length. Okay, so this is how it looks like, the gel electrophoresis. We have the cathode, which is the negative terminal, and the anode, which is the positive terminal. Okay, so that is how the DNA is separated. Okay, let's see more information. The separation is done in a sheet of gel that is about 3 to 4 millimeter thick. Okay, so we have the gel in the gel electrophoresis, of course, and in the gel close to one end are rectangular hole called wells. So which is here, the wells is here. So of course this deep blue color part is the gel, and here close to one side there is a wells here, a hole, a rectangular holes. Okay, so the wells is close to the cathode. Okay, it's close to the cathode. Of course, maybe you're wondering why they want to put in this particular position that is close to the cathode. Okay, so the reason is given afterwards. Basically, it's because the DNA samples are negatively charged, so they put more to the left so that the anode can attract the DNA sample and make it move to the right. Okay, so that's the thing, DNA molecule is negatively charged, So we put it more to the left so that they have more place for them to move to the right. They move to the right because it's attracted by the positive terminal, which is the anode. The gel is placed in a shallow tank with electrodes at both ends. Okay, so this is the tank. Okay, so the tank is here, and then there are two electrodes on both sides. And electrolyte solution is poured over the gel to cover it. So you can see the light blue part is the electrolyte. So we have electrolyte here, and then the gel is the dark blue. Okay, so one DNA sample is pipetted into each well. So you can see the micropipette is used, and they put a DNA sample in the wells. Okay, so that means one sample in each well. How many wells are there basically? From the front view, you can see only one well, right? But if you look at the top view of the gel electrophoresis, you can see actually there are more than one wells. So these rectangular shapes are the wells. So you can put some samples, DNA samples here, and then after that, they move to the other side because the anode is attracting it. So they move to the right. If you look at the front view, if you look at the top view, they are like moving down. Basically, they move to which side? They move to the anode. Okay, so why we need a few wells here? You can put samples, DNA samples. You can also put some reference with known DNA strains. Okay, so we will discuss this further. So you can put some sample and you can put some reference so that we can do comparison afterwards. So no matter how, let's continue first. Number 6, a voltage is applied across the electrodes to create an electric field. Okay, so that means we turn on the gel electrophoresis, and then the anode and the cathode have voltage, and then because of that the DNA samples are separated based on their charge together with their mass. Okay, so if they have higher charge, they will move faster to the anode. They are negatively charged basically. DNA fragments is negatively charged because of the presence of the phosphate. Phosphate is negatively charged. If you remember the formula that we learned in chemistry, phosphate is basically PO4, 3 minus, right? So this is phosphate. So phosphate is used to make DNA. So we expect that DNA is also negatively charged, but different DNA fragments will have different charge and different size. So they are separated because of that. Anode attract the negatively charged DNA fragment, and they all move to the anode, but they move at different speed because they have different mass also. Those that are longer in the DNA strain, of course we expect them to move slower, and at the same time we also expect those with the more negative charge, we expect it to move faster. And so that's why we are able to separate them. Now, number 7, these make charged molecules move through the gel because of the electric field, because of the anode and the cathode. DNA has negative charges due to phosphate in the sugar phosphate chain. So when they say sugar phosphate chain, you have to remember the structures of DNA at the same time. DNA is made of the ribose sugar, the phosphate, and the base. So there are 3 units. Okay, so the sugar phosphate form the backbone, and then the base is like coming out. Okay, so the backbone is like this, and then the base is like here. Okay, so DNA strain is basically have these structures. Now let's continue. So it moved towards the positive anode because DNA fragments is negatively charged, so they move to the positive anode. Negative and positive attract each other. The gel is therefore orientated in the electrophoresis tank with the well close to the negative cathode. So they are trying to explain it now. The well is close to the cathode so that there are more place for the DNA fragments to move and separate. So that's why the well is more to the cathode. Okay. Now after that, number 10, the gel consists of a mesh of filaments that resist the movements of molecules. So mesh of filaments is something like, try to imagine a fishing net. So it's like having certain lines to block them, so hence they are separated into different parts, so because of mesh of filaments, so they can resist the movement. Okay, so small molecules move faster than large ones, so they move further in a given time. Gel electrophoresis can therefore be used to separate fragments of DNA according to their length. When enough time has elapsed for the smallest DNA molecules to have nearly reached the anode end of the gel, the voltage is switched off and the gel removed from the tank. Okay, so which means what? Which means you let the DNA fragments to separate, they will keep moving towards the anode. When they almost move until the end of the gel, you turn off. You turn off the power, okay, no more voltage, and then they will stop moving, and then you take out the gel. Okay, so it is treated with a dye to make DNA visible. So of course we expect DNA is colorless at the beginning, we can't see it, but after that you add dye to the gel, and after that we can see a few lines. That's how we make the DNA visible. And we have a reference to be put in the one of the well also. So comparing the reference, of course, for the reference, we already, of course, the requirement for the reference is it must be a DNA sample with known DNA fragments length. We know the length of the DNA fragment, and we know what DNA fragment is inside, and they are separated. Okay? And then we can compare the distance traveled, and then from there we can see whether they have the same distance traveled or not. If they have the same distance traveled, of course, we expect them to be the same DNA fragments. So from here we can separate, and based on the reference, we can also identify. Okay, so, and from there maybe we can identify what DNA fragment is that. Okay, now the next one, number 15, the DNA has moved from the well towards the anode in a series of parallel lanes. So that means you should expect they separate like this. DNA molecules of the same length will have moved the same distance, so they form a visible band in a lane. Okay, so that's the concept, the length. If they have the same length, that means they have the same mass, and then they will move the same distance. The number of bands in a lane indicates how many different lengths of DNA were in the sample. Okay, so that means, for example, if you see there are 1, 2, 3, 4, 5, there are 5 lines, that means there are 5 types of DNA fragments. Number 18, the right-hand lane is usually used to create a ladder. Okay, right-hand lane. Right-hand lane refer to this lane, the right hand. Okay, so the right-hand lane, we put ladder. What is ladder? It's a reference. The reference is called ladder. Okay, so let's see more. The right-hand lane is usually used to create a ladder. This is done by placing a mixture of DNA fragments of known length in the right-hand well. So that's why we call it reference, At the same time, we also call it ladder. Ladder is the term that you have to use. Okay, so the ladder is used to estimate the length of bands in other lanes on the gel. Okay, so that means the ladder is a reference. You already know how long are the DNA fragments, and then we can compare the distance moved by the reference and make estimation for the DNA sample that we put in other wells. We can make estimation like based on the length moved by them, we can predict how long is the DNA fragment. Okay, so let's see another pictures here. This is an overview of gel electrophoresis. Okay, so first you can see mix agar rows and buffer. Okay, so agar rows and buffer. So it's to mix the gel actually. Okay, so you put boil mixtures in microwave. Okay, so you put this agar rows and buffer in the oven. Then after that, it is hot, it boils, and then you have to put them in the container here. Okay, so you can see number 3, cool the mixtures to 65 degrees Celsius and pour into a mold. Okay, so this is a mold to make the gel. Okay, you put in the mold, and when it cool down, it become from liquid it turn into jelly. Okay, comb insert into mold to make wells. So there are wells, right? So the wells is made in this way. So we put a comb, and then it will make the well after you remove the comb. So when should you remove the comb? Basically, when the gel solidified. There are liquid. After you cool it, it become jelly or become gel. At the time when it become a solid, gel is considered solid, and then you can remove the comb, and then there are 3 rectangular holes over here, and that is how we make the gels. Okay, so you get the gel ready, then you put into the container, and of course you can load samples into the gels. Okay, and then you can separate it by electrophoresis, and then after that, number 8, you can see after the separate, then you can put dye to make it visible. So that's why they say stain DNA fragments. Okay, so and measure distance. The one on the right hand side is the reference, by the way. Okay, the reference is known as the ladder. So they have the DNA fragment with known length, and then we plot the graph. Okay, prepare a standard curve, determine the fragment sizes. So we use the ladder to plot the graph. So how we plot the graph actually is like you look at you already know their length. Okay, you look at the distance moved. So their length is in terms of BP, that means base pair. How many base pair they have? Okay, so then how far they move. So let's say the first lane here. So let's say it has 20,000 base pair, and then the distance move is, let's say, about 8 mm. Then you put a point here. You put a point here. Then let's say the second DNA lane has, let's say, 10,000 base pair, and then they move for about 15 mm, and then you put a point here. Okay, the third one, you also know the base pair. Let's say it has, let's say I simply create, let's say it has, let's say 7,000 base pair. Then the distance move is 25 mm, and then we put a point here. Okay, so we get the curve based on the ladder. Okay, so we get the curve based on the ladder, and then for other samples, we can predict the number of base pair based on the distance move, and by reference to this graph. For example, let's say for the sample in the middle here, the first one has been moved by, let's say, 20 mm. 20 mm. If they move by 20 mm, then we look at the graph here, bring it up to 20 mm, and then bring it to the side, and then we can read, okay, so it has about 7,000 base pair. So from the graph, we can figure out this DNA sample has how many base pair. Okay, so that is how the whole thing works. Okay, then we have the fast year question here. And the answer. Okay, so this is for your own practice. Okay, now let's see the next part. So for the next part, we have the extensions of the same thing, which is about the tools just now. Okay, the tools to work on the DNA. So we applied it in different situations. Applications of polymerase chain reaction and gel electrophoresis. Okay, so remember, polymerase chain reaction is to amplify DNA. Gel electrophoresis is to separate DNA. Now we look at the application one. We can apply these two techniques in the testing for coronaviruses. A swab is taken in the nose or throat. Okay, you take a swab. Virus particles and viral RNA are rinsed off in a saline solution to produce a liquid sample. Okay, so you take a swab, then you get a sample of the coronavirus and the viral RNA. Okay, so it's in the throat swab, and then the RNA in the sample is converted to DNA. So using the enzyme reverse transcriptase. Okay, so this part maybe I have to explain a bit. Our polymerase chain reaction, right, is for DNA replication, is to amplify DNA. That means to make more DNA fragments. But the thing is, the coronavirus is not having DNA. The coronavirus has RNA as their genetic information. So because of that, in order to detect coronavirus, we change the RNA of the coronavirus to DNA by using this enzyme. This is enzyme reverse transcriptase. So this enzyme change the RNA of the coronavirus to DNA, change it to DNA for what? So that we can go through the polymerase chain reaction to amplify the DNA, and after that we can check whether the patients has coronavirus in their saliva or not. We can check that. Okay, now let's see more information. The next point, reverse transcriptase. That's the enzyme, reverse transcriptase is the enzyme. So, of course, this enzyme is to change RNA to DNA. That is what we know so far. So, it's an enzyme that converts RNA into DNA, commonly found in retroviruses like HIV. So, these retroviruses has reverse transcriptase that can change RNA to DNA. Okay, so it is used in molecular biology research to create complementary DNA strains from RNA templates, allowing for the amplifications of RNA sequences similar to DNA. Okay, so they are using the concept of complementary, base complementary. Okay, so to create the corresponding DNA strain. So for example, let's say we learned in the previous subtopics, we learned something like in RNA, we have A, G, we don't have T, okay, we have U rather than T, and then let's say C. So in the RNA, let's say we have this, and let's say in the strains of RNA, we have, in the strains of RNA, so we have A, G, U, C. Then the enzyme, which is the reverse transcriptase, will use the base on the strains of the RNA to make the DNA. So how they do it? They use the complementary concept. So they make a strain, and then after that, if A is there, so they will just pair up with T. DNA use T. RNA, they have no T, so then they use U to replace the T. So if here is G, right, then the DNA make will be C. If here is U, then the DNA may have a base of A. And then C is paired up by G. So that is how it works, from this to this, through the enzyme reverse transcriptase. Okay, so this enzyme is found in retroviruses, and the scientists take out the enzyme. You see, it happens so many times, right? Just now they take out the enzyme from the DNA polymerase of the bacteria, and now scientists also take out the enzyme reverse transcriptase from virus for their own use. So all these are biotechnology. Okay, so they take it out so that they can change the RNA of the coronavirus into DNA, and then this is the second step, RNA to DNA. and then they put it in the thermal cycle to do DNA amplification. Okay, so that's why they said it here. You create a complementary DNA strains from the RNA templates by using the enzyme reverse transcriptase, then allowing for the amplifications of RNA sequences similar to DNA. So that means the PCR will amplify the DNA. Okay, so from there we can find out whether the coronavirus is present or not. Based on the DNA, we can figure out whether coronavirus is present in the patient or not. So PCR is used to amplify specific viral-based sequences that are markers of the strains of coronavirus being tested for. Okay, so that means that PCR is to make more of the complementary DNA, make more. Okay, so why they want to make more? Because maybe in one shot, the RNA that you get is 2 liters. 2 liters for the chemical substance to detect their presence, so that's why they amplify it, make more, which means so that we can use certain chemical tests to detect whether the coronavirus is present or not. So that's why we have to amplify it first. So that are markers of the strains of coronavirus being tested for. About 35 cycles of PCR are used. So they do PCR for 35 cycles so that they can make large amount of DNA, which is corresponding to the RNA of the coronavirus. Okay, so it is this one that is amplified. So be careful with that. It is this that is amplified in 35 cycles. As PCR progresses, fluorescent markers are attached to any DNA produced. Fluorescent markers, from the name we know something. Fluorescence, something that gives light. Fluorescence is light. Okay, so a fluorescence marker is a molecule that binds to a target molecules and is used to detect it. Okay, so that means we put the fluorescence marker to the DNA. Okay, so something like this. So after you amplify the DNA, you put marker also, put the fluorescent marker here. Then after that, the fluorescent marker will release certain light, and then the machine can detect the presence of light, and from there we can see whether it is coronavirus or not from the light. Okay, so you see the level of fluorescence is monitored. So the amount of light must be enough, to prove that it is coronavirus. So that's why the level of fluorescence, which means the amount of light, must be enough, must be bright enough. If bright enough means there are a lot of these things, a lot of this. A lot of this means a lot of coronavirus genetics information there. So the light must be bright enough. Okay. If it rises above a target level, that is a reference level, if it's more than that, then the result of the test is positive. So that means the patient is infected by coronavirus. So advantages, it is very sensitive. One molecule of viral RNA is amplified to produce about 35 million molecules of DNA. So from here you can see 1 to 35 billion. It's billion, not million, it's billion, it's a big number. From 1, you can amplify to 35, so you can detect it. So minuscule quantities in a sample can be detected. That means small quantities. Small quantity in a sample can be detected. It is very specific. Primers can be designed so only one strain of the virus is detected. So remember primer. Primer will stick to certain part of the DNA and just make more for that particular part only. So a primer is a short single strain of DNA designed to bind to the DNA at the point where the selected base sequence for replication begins. So primer. Disadvantages, it requires material and equipment that are relatively expensive and are usually only available in testing laboratories. Expensive is the problem. Because of the time taken for thermocycling, results may not be immediately available. So you have to wait to get the results.